Let's jump right into this explainer. Today, we're tracking a fascinating 80-year medical detective story. We're talking about a chronological journey through key immunology discoveries, starting way back in the mid-20th century and landing right at the 2025 Nobel Prize in Medicine. It's an incredible story of pure persistence that finally unlocked one of the human body's most profound secrets, immune tolerance.
You know, over a century ago, the 1908 Nobel laureate Paul Ehrlich coined this terrifying yet incredibly accurate term, horror autotoxis. He realized our immune system has this almost unlimited capacity to destroy invaders. But without a highly precise system of tolerance, that same heavily armed system would literally turn around and lethally attack our own tissues. Figuring out how the body prevents this internal horror, well, that became the absolute holy grail of immunology.
So, here's our roadmap for this explainer. We're going to look at the immune tolerance mystery, walk through decades of Nobel discoveries, see how regulatory T cells were identified, dive into the FOXP3 master switch, and wrap up with the clinical applications and therapeutics.
Starting off with Section 1, the immune tolerance mystery.
Now, solving this century-old puzzle isn't just some dusty history lesson. It's literally the foundation of our modern therapeutic pipeline. Because we finally understand how the body learns to tolerate its own tissues, scientists have paved the way for over 200 active clinical trials today. 200! We're talking about game-changing treatments for autoimmune diseases, transplant rejection, and, of course, fighting cancer.
Moving right along to Section 2, decades of Nobel discoveries.
The path to the 2025 Nobel Prize was actually paved by a chain of brilliant breakthroughs. Back in 1945, Ray Owen noticed something wild. Calves sharing a placenta didn't reject each other's blood types. Building on that, in 1953, Peter Medawar proved that exposing mice to foreign cells in utero made them tolerate those cells later in life. They called this actively acquired tolerance, and it won Medawar and McFarland Burnett the 1960 Nobel Prize. Fast forward to the 1980s, Susumu Tonegawa wins a Nobel for mapping out how immune cells create this immense receptor diversity. But here's the catch. With massive diversity comes the huge risk of self-reactive cells. So how does the body stop them? Central tolerance in the thymus was part of the answer, sure, but researchers knew there had to be more. There had to be a peripheral break.
Which brings us to Section 3, identifying regulatory T cells.
Okay, so it's 1995, and the search for specific suppressor cells had basically hit a wall. But then, Shimon Sakaguchi makes a massive breakthrough. He meticulously separated T cells and found a very specific subpopulation expressing both CD4 and CD25, and the results were stunning. Mice that received T cells without CD25 got severely sick with autoimmune diseases, like thyroiditis. But, and here's the kicker, when he added those CD4 plus CD25 plus cells back in, it completely prevented the autoimmunity. He had literally found that elusive peripheral break. Sakaguchi named these regulatory T cells, or Tregs for short. Think of them as the essential peacemakers of the immune system. They actively suppress conventional T cells so our immune response doesn't just spiral out of control. But, science is tough. Skepticism lingered because that CD25 marker was also showing up on regular activated T cells. The field desperately needed a definitive, exclusive molecular marker to prove these cells were unique.
And that takes us to Section 4, FOXP, the master switch.
Now, this is where the detective work gets really intense. Since the 1940s, researchers knew about this mutant mouse strain, literally called the scurfy mouse, that died from severe multi-organ autoimmunity. By the late 90s, Mary Brunko and Fred Ramsdell decided to track down the cause. The scale of this was massive. They combed through 500,000 base pairs on the X chromosome. 500,000. They sequenced 20 candidate genes, one by one. And wouldn't you know it, in the very last gene they checked, they found it. A tiny, 2 base pair insertion causing a premature stop codon.
They named this newly discovered gene FOXP3. And it turns out, this transcription factor is the absolute master switch. Sakaguchi and Alexander Rudensky quickly put the pieces together. FOXP3 expression exclusively characterizes regulatory T cells and ensures stable immune balance. It's the single gene that programs a regular T cell to become a protective Treg. Without FOXP, you literally have 0 Tregs, and your immune tolerance completely collapses. But this wasn't just about mice. Brunkow, Ramsdell, and their clinical partners immediately looked into a rare, devastating human disease called IPEX syndrome, which causes fatal autoimmunity in young boys. And they found that the exact same genetic flaw, Mutations in the human FOXP3 gene was causing IPEX. It was the exact same mechanism. Both the scurfy mouse and the human IPEX patient were suffering because they lacked these regulatory T cells. To absolutely seal the deal, Brunko and Ramsdell did these incredibly elegant genetic rescue experiments. They took the healthy, wild-type FOXP3 gene, introduced it into fatally sick scurfy mice, and it completely cured them. It was total rescue. This proved beyond a shadow of a doubt that a single gene locus, FOXP3, controls the exact cell type, Tregs, that we need to prevent that horror autotoxicus.
Let's head into our final part, Section 5, Clinical Applications and Therapeutics.
Bringing it back to the clinic today, we are looking at over 200 clinical trials actively trying to harness or modulate regulatory T cells. Because we now know that FOXP3 expression characterizes these cells and locks in that immune balance, we can actually engineer them. For the healthcare leaders tuning in, consider this your executive briefing on the pipeline. It is packed. We're looking at taking a patient's own Tregs, expanding them in a lab and giving them back, or even engineering CAR Tregs with chimeric receptors to target highly specific sites of inflammation, like in bowel disease. There are TCR Tregs for things like celiac disease. We're even testing low-dose IL-2 agonists to expand Tregs directly inside the patient to treat atopic dermatitis. And, on the flip side, we're developing targeted antibodies, like against CCR8, to selectively wipe out the Tregs that are hiding inside tumors.
So it all comes down to this ultimate clinical goal, mastering the therapeutic balance. It's like a giant seesaw. On one side, for patients with asthma, allergies, autoimmune conditions, or organ transplants, we absolutely want to boost Treg function to restore tolerance. But on the other side, for cancer patients, tumors actually use Tregs as a shield against the immune system. In those cases, the goal is to selectively deplete those tumor-infiltrating Tregs, basically cutting the brakes, so the immune system can unleash and destroy the cancer.
I'll leave you with this final thought. The discovery of regulatory T cells and the FOXP3 gene by Brunko, Ramsdell, and Sakaguchi, our 2025 Nobel laureates, has fundamentally rewritten the rules of immunology. As these targeted therapies rapidly make their way from the lab bench to active clinical use, mastering immune tolerance isn't just some scientific mystery anymore. It is the absolute future of patient care. So the question is, how is your organization preparing for the upcoming Treg therapeutic revolution?